Thank you for the opportunity to present as part of the symposium. My name is Catherine Wedderburn, and I'm presenting with my colleague Kirsty Donald, who will be there in person. And our presentation is on the association of environmental factors with brain structure and function in a South African setting. I have no disclosures. I'm going to start by discussing why it is important to examine brain development in low and middle income country settings. And on the graph on the left, you can see the predicted population growth from 2015 to 2100. And what is immediately obvious is that the population of Africa is rapidly growing. And on the right, what we can see is the global population demographic is evolving with changing socio-demographic circumstances. And in the global north, people are having longer lifespans with resulting higher numbers in the older age range. Whereas on the other side, the African region remains in population growth. And the population prediction is that Africa will host nearly half of the world's children by 2100. And therefore, optimizing development of children in Africa is particularly important, both at an individual level and at a global level, as Africa will be a huge source of the global population by the second half of the century. However, children in this region are at the highest risk for not reaching their developmental potential. And from the map, you can see that Africa and Southeast Asia are disproportionately burdened by neurodevelopmental disabilities, driven by environmental risks, so higher substance use, infectious disease exposure, poorer nutrition, mental health, and conflict situations, but also reduced access to care. And when we look at brain growth across the lifespan, we can see that the most rapid changes occur in these early years. And these lines all represent different aspects of brain development. And if we look in the middle, that's brain volume. And you can see the changes from antenatal birth through to 100 years. And what we see is that 80% of brain growth happens by age 3. And given the critical importance of the first years of life on later cognitive and health outcomes, it's really important for us to understand what influences this early brain growth, and particularly in low middle income country settings and Africa. So what have we found? The Drakenstein Child Health Study is a South African population-based birth cohort that is investigating the early determinants of child health and development. And it's located in South Africa outside Cape Town, and it's shown there on the map in the red dot. And Professor Heather Zaar is the PI of the study, and Professor Dan Stein leads the psychosocial arm. And the study recruited women in pregnancy and follows up mother-child pairs through adolescence. And these families live in a peri-urban region with low socioeconomic status, high risk for substance use, infectious diseases, and other risk factors. And a subgroup of 220 children in the cohort have undergone longitudinal neuroimaging at 2 to 6 weeks, 2 to 3 years, and 6 to 7 years. And we've included various modalities, but I'm going to focus on structural and microstructural results for this presentation. And we've also conducted clinical neurodevelopmental assessments of these children. And just to be able to visualize the study setting, the pictures on the right are from the local community, and the left is the hospital in Cape Town where neuroimaging takes place. And we conducted neuroimaging without sedation. And when they were younger, this meant we needed the children to fall asleep in order to lie still for an hour of scanning. And as you can imagine, this has been very challenging to set up, both for the researchers and the families. But we've really honed a family-friendly approach. Happy to share more details. And you can see one of the happy children here in the scanner. And we've been very successful with this and have got some very interesting results. And the first thing that we've done is to look at the association between brain structure and function, specifically cognition and language. And these are the results at 2 years of age. And we've mapped specific regions that are associated with different functions. And this has then gone on to inform our analysis of risk factors. And the risk and protective factors are really a complex web that influence this early period and then ongoing. And their brains are growing really rapidly over these preschool years of life, which means that issues that start out in early life may become more manifest over time. And for the purposes of this presentation, I'm going to focus on a few of the factors from our setting and the associations with child brain structure. And for each factor, I will speak to the importance of this and then briefly describe our results to date. And I'm going to start off with maternal depression. And this is a map of the rates of postnatal depression estimated at 17% worldwide. And although there's a lot of data missing from Africa, there are very high rates reported in Southern Africa of around 40%. And we've examined this in both the neonatal child brain structure and the 2 to 3 year old age point, which you can see here from the figure. And we've seen an association between maternal depression and child brain structure, specifically the amygdala, at both time points. And at the 2 to 3 year old time point, we noted particularly an impact of postnatal depression and persistent exposure. So maternal depression across pregnancy and the postpartum was associated with altered amygdala volumes. And what's interesting about this is this region is known to be involved in socio-emotional behavior, which suggests a potential transgenerational risk. Next, examining anemia, which is a global issue and is known to be a risk factor for child neurodevelopment, but is particularly prevalent in sub-Saharan Africa, meaning we have an opportunity here to answer questions of global relevance. And we've looked at both anemia in pregnancy and anemia in children and the relationship to child brain structure. And when we looked at maternal anemia in pregnancy, and there was mild and moderate anemia, but actually not severe anemia, we found an association with smaller caudate and putamen volumes and also smaller corpus callosum by 8% when compared to children born to mothers without anemia in pregnancy. And interestingly, we did not see an association with child anemia, which indicates the importance of the in utero period, and that there therefore may be these sensitive windows where if you can intervene, you can prevent later poor outcomes. Another risk factor we've looked at is prenatal alcohol exposure, which is prevalent, as you can see on this map, in a number of places. One of the key regions where this question can be answered alongside other environmental risk factors is South Africa. And for this, I'm going to present the results of the diffusion tensor imaging longitudinal analyses. And we found that prenatal alcohol exposure was associated with differences in specific tract maturations, notably the superior cerebellar peduncle and the superior longitudinal fasciculus, suggesting altered white matter development among children with alcohol exposure, which has importance for various aspects of neurocognitive function. And this study highlights the value of repeated early imaging because we've seen these differing rates of maturation across the tracts. Finally, I wanted to describe the findings of HIV exposure. So this is a regionally important public health problem as antenatal HIV rates remain high in sub-Saharan Africa. But thanks to antiretroviral therapy during pregnancy, the rates of vertical transmission have decreased, such that most children born to mothers living with HIV remain uninfected. However, given the very large numbers of children, so 1 in 5 children born in South Africa are HIV exposed and uninfected, it's important to examine if there are any effects of HIV exposure on child brain development. And we looked at both neonates and 2 to 3 years, and these are the 2 to 3-year-old results. And we found that these children are at risk of poorer language development compared to HIV unexposed children. And we found an association with cortical thickness in the medial orbital frontal region. And when we put this together, we found that the cortical thickness, the effect on cortical thickness mediated the association between HIV exposure and poorer language development, suggesting possible underlying pathways between the structural and function effects. Now, we're still working to understand the significance of these findings and long term, and we need to monitor these children to understand what is going on, how they interact, and how best we can therefore intervene to optimize potential. Finally, I wanted to highlight a new study that is being conducted examining low-field neuroimaging. Now, one of the challenging aspects of examining brain imaging in Africa is access to MRIs. And this is looking at how we can use low-field scanning in these settings. So please watch this space. I would like to acknowledge so many people, institutions, and funders, particularly the families of the Drakenstein Child Health Study. Thank you very much for your attention. Please feel free to contact me or Kirsty with any questions.